Hi, everyone. Ben here again. I'm going to talk today about returning to the questions and thinking about what the next set of models are to address those questions and some ongoing developments that I think are of interest moving ahead in the next couple of years, thinking about human genetics. And if you remember what I talked about from last time, you'll remember that we have generated as a community a large amount of genetic variation associated with complex traits and phenotypes that through that discovery process, we've really started to answer some fundamental questions about the nature and structure of complex traits generally. And like any good scientific question addressed, that leads to many new additional questions that need to be addressed. And ideally, and the whole reason we're doing this is because we're keen on trying to understand biology, mechanism, and ultimately use that information to return to the clinic and help people. That was our stated objectives 20 years ago. Those objectives still remain potent today. So if you remember from last time, I suggested a range of questions, and I've just brought this slide back up again, reminding us that there are many, many unanswered questions after a GWAS. That experiment is great to identify regions of the genome that are associated with the trait beyond doubt, but it doesn't tell you necessarily at face what the causal gene is, the variant, the horsepower, the direction of effect, what the mechanism is, what the therapeutic modality, directionality is. So we definitely need to do additional work to try to understand these questions better. One interesting thread and idea, and I definitely like this idea. Uh, secretly I'm probably a Bayesian, but up front I'm mostly a frequentist. But as a Bayesian, secretly, we all have priors, and we're all trying to use information and data to try to educate what we think the right models are. And the best way to educate your priors is with data. And perhaps one way we can start educating our priors is to, thinking about complex traits like diabetes or schizophrenia or autism or Alzheimer's disease. These are pretty complicated traits at the clinic to figure out and genetically dissect. You need lots of samples, lots of data. And even when we do that, we don't fully understand the architecture and we need more work. So what if we instead approach the understanding of architecture through other types of phenotypes, not physiological traits, but actually like molecular phenotypes? Maybe the patterns of transcript abundance, gene expression might be one way in which we can understand just generally the architecture of a trait, if that trait is just expression, and then and the process of mapping genetic variants associated with expression, we can use that information to understand how regulatory architecture works, understand the relationship between all of those genetic architectural parameters, like how many loci are involved, what's the allelic spectrum, what's the frequency of those alleles, how many alleles per locus do we expect. And we can build, by doing that successive mapping, we can build a better foundation and understanding about what we think that right architectural models are, and then use that insight to try to guide what we expect to see through, sort of like by analogy to complex traits. So if you're not familiar with, gene expression mapping trait variation, almost surely in previous lectures you've seen this, but very, very briefly, this is a very simple idea. And again, models the same study design in GWAS, except in GWAS, rather, in that case, you have a trait like height or schizophrenia or diabetes, and you're trying to physiologically measure that in patients, and you're trying to correlate the change in dosage of a genotype with that change in outcome trait in that very, very simple regression model. Well, so in the expression case, you're just swapping out the physiological trait and swapping in a measurement of transcript abundance, perhaps collected by RNA sequencing, for example. So in sort of like our, analogy here on the left, you've got your disease risk from some variant where you can see that increasing dosage of the T allele increases your disease, but you can also see that increased expression, increased dosage of T also increases expression of this gene. And we can, the dream would be that we can do this sort of like linkage between this genetic variant and the expression of the gene and then ultimately mediating the risk of disease. That would be sort of like the dream case that we could have. But in this analogy, you can see that, oh, well, we can map genetic variants for expression, and that sort of like can start to tell us a little bit more about architecture. So, in principle, maybe because we don't yet fully understand the spectrum of variation associated with traits, let's do something a little bit simpler, build our understanding of the architecture there, and actually in the process of mapping those genetic variants associated with gene regulatory architecture, maybe we're just lucky enough to like make linkages to disease traits as well, and then we can kind of get a twofer and learn about architecture and try to understand and diagnose disease better. So great projects and a great set of experiments to try to think through. I wasn't a member of this, and you've probably heard this from other sources, but I think one of the projects that sort of like leads in this effort is sort of like the GTEx project. And that was a project where they collect donors, measure transcript abundance across a range of tissues, across many, many subjects, and then simultaneously collect genetic variation data attached to it. So in that design, you now have the ability to measure the relationship between transcript abundance in all of these different tissues against genotype, or genotype against transcript abundance. So that allows you to map those variants that are associated with that change in transcripts, and we can do this at scale. And it turns out the power for discovery of this particular molecular phenotype is much stronger. You need far fewer samples conventionally than you would to map sort of like small changes in effect sizes and odds ratios for other complex traits or other disease traits. So this design allows you to just like systematically and at great scale map genetic architecture for genetic variants associated with gene expression very, very readily. So what I think is really nice, again, when you're wearing this cap of like, oh, we've done that experiment, what are kind of like the large scale take home messages that we should take back, thinking then about what we're going to be up against when we're really thinking about mapping complex traits. And then for those places where we're like, oh, well, we definitely see that this is going to happen, we should probably gear up our human genetics experiments for those physiological traits with that idea in mind. And so what I'm going to just go walk through very quickly, just some, from my point of view, some really key take-home messages from that set of experiment, sort of like enumerated here, but I'll go through each of these one by one. So one of the obvious things is that there are a ton of regulatory variants to be discovered. With this design of, upwards of 900 800 subjects with variation in the total sample numbers varying by the tissue that was collected. Here you've got plotted sort of the number of genes that have a genetic variant associated with either change in transcript abundance, so-called E genes, or change in alternative splicing, splice QTLs, as it were, or S genes. And what you can see from this is that this plot is relatively linear with respect to E genes. So what that means intuitively is that we have not saturated the distribution of variation associated with those traits. If things had sort of like bottomed out, then we'd be like, oh, okay, well, we're kind of like capturing most of regulatory variation and it's sort of like capped out. But, as we increase the sample sizes, we just discover more things. And intuitively, that kind of makes sense. We're probably extremely well powered to find regulatory effects that have a large effect, much less powered to find things with weaker effects, but for sample size proportionally, we can increasingly penetrate that space, find effects that are weaker, or test rare variation that has stronger effects. So in this sort of like power curve, there's this relationship between power and frequency and effect size. So in that interface, we can discover things readily, but we're still not fully capturing everything, and there's still a lot to be found. Another obvious and very straightforward thing is that component of architecture, that allelic series that you find at sites, is a common feature. It's not an exception. It's probably the rule. So if you plot the number of EQTLs regionally per E gene as a function of sample size, what you see is that the proportion of those sites per E gene just goes up as a function of sample size. So, and it's linear. So again, we haven't hit the saturation threshold where, oh, we increase sample size and we barely find additional signals. That's not what happens at all. You increase sample size regionally, you just keep finding additional variation with weaker effects associated with that same E gene or E genes regionally. So we are still sample size limited in our discoverability, and we just keep finding more variation regionally that's associated with change in gene expression. So that makes intuitive sense. Again, many, many models in the Mendelian monogenic form match sort of like the intuition for this. And so again, implies that we should probably work hard to identify all those variants. And to do that, we're going to have to use other strategies to do signal find mapping and so forth to discover this variation. Of interest of late and increasingly interesting is to try to map cis-regulatory elements. So that's the place where you've got some regulatory enhancer or repressor element, and you know that's attached somehow to a cognate gene of which that element is intimately involved in regulating expression. And you can imagine genes are expressed in individual tissues and multiple tissues, or maybe even ubiquitously. And you can also imagine that there's also multiple regulatory elements. There's not a single enhancer or a single repressor element involved in the regulation of that gene. You can imagine all the reasons why that could be because of timing, because the regulatory architecture is complicated, it requires circadian rhythms, tons of things that could be involved in that. And so again, when you start to map genetic variation associated with regulation and then try to attach cis-regulatory element architecture through other means, what you find is that in practice, first off, that those sets of variants definitely overlap cis-regulatory elements. And in fact, so that makes sense intuitively, biologically. We'd hope that like a gene that changes expression that maps to some enhancer regulatory element is in fact attached to a gene and so connects with the biology we intuitively understand about that. So that makes sense. But actually, more interestingly, that cis-regulatory architecture is reused and used in multiple cases. So it's not just a single element that hits a single gene. You get multiple regulatory units intersecting the same element. And so the overlap between those is actually more frequent. So that has some pretty important implications. It probably means there's a lot more pleiotropy than we probably are understanding, because a gene can do lots of different things, and a regulatory element, when it's disrupted, might lead to changes in that gene in a bunch of different contexts. But also trying to map down to the precise variant to gene to function to phenotype may be more complicated. If an element has 4 CREs attached to it, it might be that only one of them is really truly disease relevant in the context of the disease you're studying. The other 3 might be spurious. So trying to sort that out is going to be much more complicated. So biologically realistic, but it definitely is going to force us to think harder about how we understand this relationship structure. It turns out that QTLs are both, eQTLs are both specific to a given tissue, but also are expressed in multiple tissues. So the number of tissues that are shared sort of have this sort of like bimodal tail distribution. So you see many cases where as sample size goes up, you can discover effects that are very specific to a tissue, but then also are generalized and are shared across many, many tissues. And then if you look at the effect sizes as a function of the number of tissues that gene is expressed in, what you see is this subtle decay over distance. So the stronger effect sizes tend to be attached with genes that are expressed in a single tissue or fewer tissues versus those that are, the effect sizes are weaker when the tissue expression is much more plural. And that could easily be understood in the context of population genetics and selection. A gene that's expressed in a lot of tissues probably is one that's important, and you can't tinker with population genetically too much, because if you do, it'll probably have more background deleterious consequences relative to those that are kind of tissue specific, which it's still perhaps bad to tinker with it perfectly, but you might be able to, a stronger effect sizes might still escape the full gaze of background selection, and some of those variations are allowed to persist. There's lots of lessons that we want to learn from sort of like integrating that data together that we're going to have to think through. So thinking about the frequency of those effects, how many SNPs are involved, how many SNP to gene connections, and what tissues those are involved in, because the regulatory architecture is just way more plural than it is singular. It definitely is going to increase the challenge of us sussing out the specific relationships between gene var you know you know genetic variation, change in dosage to mechanism to functional element to gene to tissue to physiology. So one thing that, of course, immediately falls out, as you might anticipated, given that there are so many QTLs to be discovered, if you just picked a random QTL, random SNP in the genome, it is almost surely nominally significantly associated with some variant in the genome that's actually an eQTL. But that variant itself is not probably an eQTL. It's just partially in LD or in weak LD with some variant that is actually a cis eQTL in the genome. So just enumerating that, like, oh, is a variant associated with regulating gene expression? Oh, yeah, certainly it is, just by chance because it's tagging that. Itself, it may not be causal for it. So what that means is that we have to do a much more fine scale, high resolution job of mapping those causal variants for change in gene expression or change in molecular phenotypes so we can attach specific causal variants for those to either a gene of action or to a physiological trait of action. It's just not enough to just look up a p-value for association that way. So you probably talked about this in co-localization experiments, and the toolkit to do that is just one of these like fundamental things that like transforms how you think about mapping and understanding pleiotropy and kind of like making connections between handling the issues of linkage disequilibrium that mean that we don't perfectly tag causal variants, but we're looking for like common signals across them. So here I'm just giving a very, very short primer of co-localization. Hopefully you've seen that in other places. But if you haven't heard of this framework, it's very, very straightforward. The question you're trying to ask is, you've got two sets of, trait mapping studies. Let's imagine in one case, you've done your GWAS for diabetes or schizophrenia, and you've got your plot of the association by physical position on the x-axis, and then the association p-value on the y-axis. And you've got that for type 2 diabetes or schizophrenia. And then you've got, oh, I've got my eQTL data. So I've measured the association between genotype dosage and change expression in my favorite gene that turns out to be like a gene in a tissue type that's relevant. And perhaps I luck out. And in this case, there's perhaps a signal of association with that transcript abundance nearby. And maybe it's like close to, or maybe even right on top of the same signal that my trait is associated with. And I'm like, oh, I really would like to know if that sort of like initial mediation model in that, first or second slide that I showed just previously, where you say, oh, I've got a variant that changes expression of the gene nearby. I have that same variant that changes the association with my trait. So can I draw this like mediation model between SNP and exposure of exposure of transcript abundance to outcome, the association to my trait. So co-localization is a framework that allows you to sort of like make that connection. And if you think about the possible configurations that you might have, those are kind of laid out here in pictorially on the left axis here. So just to be fully transparent, there's many, many possibilities. So like one possibility is that you have no association either in trait 1 or trait 2 in the genome. That's a sort of global null hypothesis. That's sort of like boring. You probably wouldn't even try to line those things up if you didn't have that information. There's two other cases where perhaps you've got an association in one of the traits or the other trait, but not in both. And so, okay, well, that's easy because there's no signal, so there's no need to try to co-localize. But it's like this H3 or H4 hypothesis that are more interesting. So you've got this region, you've got evidence of association for trait 1 and trait 2, and you want to distinguish between those cases. You want to know, is it association proximal, but the signal isn't shared because it seems to be tagging different causal variants? Or is the signal in the H4 case overlapping, and you can see, oh, the association evidence seems to be tracking together. The strength of association correlates, and the strength of that relationship is evidence for co-localization, that in fact the same causal variant is contributing to trait one and trait two. So this co-localization framework gives you a probabilistic Bayesian form to try to assess each of the probabilities of each of these configurations given your locus zoom plot or your association data regionally. And it's just a great easy way to distinguish in that, sort of earlier case, which is like, oh, I have a nominal EQTL association, but is actually mechanistically, are those things linked together? This is what co-localization can help you do. So when you take that framework to a collection of traits and you map with GTEx data, what do you find? Well, it turns out that this experiment works reasonably well. You find many examples where change in gene expression does co-localize with one or more physiological traits, cholesterol levels, body mass index, Crohn's disease, all sorts. And even to some extent, you might have said, well, could it have been the nearest gene? Like the nearest gene may be the right gene, so it's a pretty good guess. And it turns out in some of those cases, it actually is the nearest gene, and so that's pretty good. So you get co-localization, it happens to be the nearest gene, but also in some cases, it's actually you get a strong co-localization, but it's actually not the nearest gene at all. And so you might have guessed wrong if you were like, oh, my gene variant is like right on top of this gene, maybe that's the right gene. Well, maybe regulatory architecture doesn't know boundaries in that way, and it happily can have a long distance enhancer that like loops to a promoter of a distant gene, and you didn't know that because you didn't have the data to make that linkage. Co-localization in this kind of experiment can help bridge that gap. But one thing that also falls from this is that, in fact, not all variants co-localize for these traits. So EQTLs, as in the sort of like regulatory architecture, explain some fraction, but not the entire fraction of things. And I think this leads to a interesting conversation in the field that's currently ongoing, which sort of is this, analogous to historically us thinking about what was the missing heritability gap. So in that case, it was like, oh, we did this experiment. We said, oh, common variants can probably explain heritability. We do the common variant experiment. It explains some fraction, but not all of the heritability of the trait. And we're like, oh, what's the missing heritability gap? What are the causes and sources? Well, here it's an exact same analogous scenario. We see this gap in the co-localizations that occur. And so that's led many to think about what's causing the co-localization gap. If we believe that ultimately gene regulation is the cause and source of the majority of the associations that we find from these traits, then EQTLs would have been the obvious thing that should have explained a lot, but because it doesn't, leads to other ideas about what might be the sources of that. And just to give you a hint of, perhaps some of that gap and what it could be that EQTLs are correct, but we're not capturing all the EQTLs because we haven't surveyed all common and all low frequency variant QTLs that map effectively. We haven't saturated that power curve. So maybe if we doubled or tripled the sample size, we would find EQTLs with weaker effects, and then those weaker effect EQTLs would co-localize. That's a possibility. We are only measuring EQTLs at a single state and a standing state at a particular time point, and certainly are not dynamic in all the ways you can imagine that dynamicism. So there's not a perturbed state where we know that like QTLs can have effects with different environmental conditions or stimulation. Start to think like immune signaling is one way, or like, oh, if you sense this signal in the data like cortisol, that might change how you have expression, but only in the context of that perturbation might you see a QTL. So dynamic, not static QTLs. So that could be a possibility. Maybe it's not EQTLs. Maybe there are other QTLs that we're better able to map, like variation associated with chromatin accessibility, which I'll talk about in a minute. Maybe that's actually a source of variation that we also think is attached to expression, but maybe we're slightly better at mapping those variants for various reasons because we can capture some more dynamicism. So lots of different places where this could be explained better. And I don't think there's going to be even one answer to this. I think there's lots of answers possibly to this, but I think the field is like trying to figure out exactly what's next to try to solve this. But this gap is something of interest to many. And just to hit this home, when it does work, it works well and actually can really just like hone in on all the things that you'd really hope when you're trying to like dissect all the questions that we laid out. So probably like the canon of examples among many examples, and this one's, I'm relatively familiar with, is the sortilin 1 locus. So this is a locus, one of the strongest signals for association with LDL cholesterol. It's off the charts for that. So genetic variation here definitely associates with LDL cholesterol. Because we know LDL cholesterol is a risk factor for coronary artery disease, it's not a surprise that that same variant or that collection of variation, tag variation, also associates with heart disease. So that makes a lot of sense. But then you want to know what's the causal gene and can we figure and suss out those details? Well, in this plot, in liver, you can actually see that there are associations with that same variant associated with heart disease, and a number of genes nearby in cyst associated with gene expression, including CELSAR2, PSRC1, and sortilin 1. So makes a lot of sense. Once you drill down on that, you can see actually that there's like a consensus binding site for CBPA, which is a known active regulator in liver. And so, and the variant seems to disrupt a particular binding motif for that transcription factor. So all that makes sense. And there's definitely tons of work sussing out that relationship structure and the causal variants exactly. So this all very, very, very much makes much sense. I'm not going to tell you the whole story about this, but we think sortilin 1 is the right gene. Even in this case, if the EQTL data is suggesting that sortilin 1 and a couple of other genes seem to also be coordinately regulated by this particular transcriptional, this collection of genetic variants, presumably through this particular genetic mechanism. But we don't necessarily know from these data that which one is the causal variant itself. So again, EQTLs are great. They may not be perfectly salient in solving all of our questions, but can kind of help hone our experiments to kind of like rule in leads for functional experiments. So let me just transition, in closing to just thinking about other thoughts about where I see cool trends in the field that I think are going to pay off quite substantially. And I start about this thinking about what we're going to do in the future, just reminding ourselves that we approach our development of technologies and the data and our models thinking very I don't want to say, we had very high dreams of simplicity. And that was not unreasonably based. We were drawing our intuition about what we thought we might find in complex traits based on all the successes we had in linkage studies and linkage mapping analysis, where it turned out those rare diseases were by and large single monogenic causes where the patterns of inheritance were relatively straightforward and sort of like, reified our intuition from our basic genetics 101 classes, thinking about P segregation and autosomal dominance and transmissions and all these sorts of things. And now as we've moved in this era, we sort of like recognize the idea that like complex traits are complex for sure. And this components of architecture are ways of establishing a framework to understand that complexity and ways to try to dissect it. But we should not assume that all loci and all traits have the same amount of complexity and they are equal in that regard. Some traits and loci might be simple, other traits and loci may be more complicated. And just thinking about that at the genome-wide side or locus architecture, just sort of like this is like a pictorial representation about one way that I've been thinking about this. But you're thinking about genetic heterogeneity going from monogenic to all the way to omnigenic, where almost everything is implicated, to the frequency and effect size relationship, where you've got sort of high effects that are de novo to common that are that are high frequency but weak effect sizes. Within the region of the locus itself, you may have multiple independent signals. Within a signal itself, you may have a single causal variant, but you may have multiple causal variants that are implicated. You don't have to have one gene implicated. It might be that you have regulatory architecture like operons that sort of mean that one regulatory element impacts the expression of multiple genes, and all of those genes or some fraction of them are physiologically relevant. You don't have to have the same mechanism. In fact, it may be reasonable that multiple variants impact the gene in different ways, but in consistent ways, or maybe even sometimes complicated ways. A coding variant might, a missense variant versus a loss of function variant might do something different to the protein than a splice altering form that's loss of function, or exon skips versus one that simply changes gene expression of all the transcripts, or a subset of the transcripts by some small amount. So those mechanisms may do things differently to the phenotype. And we should be aware that that can happen, and that could have downstream effects on how we think about it, how we think about dissecting the trait. And ultimately, the tissues themselves, it could be that a single tissue is implicated, but it definitely doesn't have to be that way. It could be the gene, and the effect has implications in multiple tissues. And so trying to suss out the increasing complexity across all of these axes is definitely, I think, one of the more important challenges and harder challenges in thinking a little bit into the future. I think you can't solve all of those problems, but what we can do is try to, all at once. But what we can do is do what we always do, which is like smart people picking particular problems and trying to figure out statistical, technological solutions to those challenges. And then once we have them, really just deploying them at scale to really try to get as best and deep an information and resolution to that question as possible. So one of those approaches, as I've alluded to, is thinking about identifying causal variants through signal-find mapping. And so that's that approach where you've got your region of association, you've got your lead variant, you think there's interesting action going on. You'd like to know, is there one signal, or is there a series of signals that are distinguishable and independent of one another, that both coordinate action at the locus? And if so, what are the set of variants that are capturing each of those signals that you're credibly, statistically think are most likely to harbor one or more causal variants in them? So this signal-find mapping effort is important. And we actually today have probably the best chance and best hope to do this experiment at scale and do it really well, because we're armed with the best tools we've ever had to dissect this. We were armed with the best data sets that we have ever had through DNA biobanks and other sources. It's not just one biobank, it's many. And you can realize right away that signal-find mapping, the power of this experiment, is really intimately linked to how much data you have, how many samples that you have, and how well those samples are phenotyped, and how diverse those samples are. And it can't just be one group doing it with one biobank. It's got to be accumulation of all the data across multiple biobanks. So thinking about how to integrate that data together to do that. And so methods development space in this space has been very, very active in the last couple of years, with really transformative tools that really have made this sort of possible, and kind of like are capturing a couple of key features. They're working with summary data itself, so not at the individual level, and capturing appropriate LD reference panels that match that summary data, so you're not worried about mismatch between the patterns of LD you're using to model the association data and the set of individuals that were used to generate the association data. If that's perfectly, nicely, tightly matched, then you're getting the best approximation to the actual experiment at the individual level. And remember, that's important because you don't want to do this analysis in one biobank. You're doing it in like 10 or 20 biobanks, and you're trying to bring all that data together in a single place. And if you had 20 people doing it, it would be an impossible experiment. But having one person or a few people collect the data from other sources and collect and accumulate it, that makes this experiment possible and feasible and transformative. But these tools can work with that data, can analyze with different patterns of LD, can capture differences across ancestries, can define credible sets likely to be associated. Maybe you could integrate functional variation, so like chromatin accessibility or other types of data, to further narrow the credible set of variants you think are statistically likely. Maybe even could use multiple trait data where you're excited by, oh, well, maybe there's a site that's associated with type 2 diabetes and BMI, and it looks like it's the same signal. And if it is, maybe I can even use those trait data to narrow in on the signal more. But the tools to kind of like utilize all of these kinds of different informations are definitely growing and available, and are really giving an opportunity to like do this experiment at scale and really nicely. And the multi-ancestry component of this is really, really important. And you might say, well, why is that important? The fundamental reason is that different populations have different demographic histories. Some populations are just simply their history is older in evolutionary time, which means that recombination has had a longer period of time to act. And so the LD blocks of those so around those association signals, because more recombination has happened on average, those blocks tend to be a bit narrower. And so for signals that are shared across different groups, you can more quickly hone in on the right causal variant by integrating the data across multiple ancestry groups. And so here's like one example from this from Graham 2021, and I think this is about LDL cholesterol. So this is like a signal of association for cholesterol levels. But what you can see is that in one case, the signal in African Americans really hones in on a particular variant. And if you look at other ancestry groups like Europe, you can see that same variant highly represented, but carried along with it are many, many variants in LD. And so if you were trying to suss out the causal variant, you would credibly say, oh, well, it's probably the purple one plus maybe some of the red ones, not sure. But when you look at and integrate across ancestries, you can make the case that, oh, perhaps it's this lead variant here. That's sort of like the signal by parsimony gives you sort of like credible evidence that that's the correct variant. So using those LD differences can really help improve that fine mapping resolution. And can really help that. And these experiments to sort of like capture multiple ancestries to do fine mapping at scale are definitely happening. So this particular study, a preprint, I don't remember if it's published now, but definitely changed my mind and influenced me about how I think about this problem. This was a very, very early stage of collecting 3 different biobanks of data, not completely representative of diversity, but partially represented. And immediately when you do this experiment, you definitely find all of the things that you would imagine finding, given what we previously talked about just a couple minutes ago about architecture, mapping genetic variation associated with gene regulation, EQTLs. You find multiple signals per site. You can narrow those credible sets down to a certain size. You can even find individual variants that are highly likely to be the causal one. And you can connect those signals with QTL variation at scale. And this just works. And so the question is, because this works so well, we should just scale this up to as many samples and biobanks as possible. And again, this is work from a collaboration that I've been involved in. Really trying to take that at even further scale. So not just focusing on like a handful of traits, but expanding the ancestries and expanding the phenotypes at study really allows you to do this fine mapping experiments at scale. And again, what you find is, very, very straightforward things that you can actually do fine mapping. You can narrow down credible sets of association down to a handful of genetic variants, that most of the associations that you're fine mapping are driven by power. So in MVP, for example, we have representation from Europe, African Americans, Hispanics, and a lesser extent, Asian populations, but the lion's share of the sample size is from individuals who are similar in ancestry to 1,000 genomes European populations as a reference. And so there you definitely see many of the signals, but you also definitely see signals in these other groups, continental groups that look like African or American or East Asian 1,000 genome reference, similar populations. And where you may only see the signal fine mapping in one group, mostly it's due to just power issues. So it's not like there isn't a signal present in those groups. You just don't have a potent enough signal to do the fine mapping experiment. So what does that imply? It probably just implies that we need to assemble the largest cohorts of data for all the different ancestry groups and try to pull that together to do the fine mapping experiment. And if we do that, we'll be able to further narrow the resolution, find signals of association, map allelic heterogeneity, and really learn this experiment and the architecture of allelic heterogeneity, locus to locus, quite at scale. And this is just like one experiment. I know that this experiment is being further extended at scale beyond a single biobank to multiple biobanks. So an exciting time to see how this plays out. But aside from fine mapping, I think, returning to that question of like that missing co-localization gap, could it be that there are other sources of molecular phenotypic variation that could help to explain possible mechanisms that contribute to association with complex traits? One of those that's very, very obvious and straightforward is chromatin accessibility. So those are the places in the genome. If you know, think about your gene regulation biology 101, you might remember that DNA is compacted, it's wrapped around histones, and there are places where those histones have to, expand and contract to allow naked DNA to be exposed so that transcription factors can bind and organize and promote transcriptions of genes regionally. And that accessibility of chromatin is definitely a key feature in the appropriate regulation of genes nearby in cis. And so the idea of mapping regions of accessible chromatin and is fundamental to understanding gene regulation just at a basic level, but then further mapping genetic variation that's associated with change in chromatin accessibility level by profiling chromatin accessibility and doing that in a population of people, and then saying, does change in genotype dose associate with change in chromatin accessibility, gives you a different molecular phenotype that also tells you about regulatory biology, a different set of things than you might get from EQTLs, but maybe a type of molecular trait that's easier to map, maybe more robust, and tells us more about the plasticity of gene regulation than EQTLs do. And so a couple of cool experiments that, that I've been involved in and that I'm aware of that I think kind of telegraph this is in one experiment, getting CAQTL data is not straightforward. So perhaps, but a lot of folks have generated chromatin accessibility data as profiled by ATAC-seq data. So what you could do is go to the resources that have already been generated, the public resources, collect all that ATAC-seq data. Those data they weren't, the people that generated all those data weren't thinking about mapping variation associated with change in chromatin accessibility state. So there's no genotype data. But what if you could attach genotype data to it? And the clever idea to do that is to actually just use the reads that were generated from the ATAC-seq data to map genetic variation, because you've profiled reads, you've mapped the reads to the genome, and you can find some of those reads align perfectly, some of those lines map imperfectly, and you can see the reads generate genetic variation, and we know a lot of genetic variation already. So that's not like a great profile of genetic variation. You're not getting like great coverage across the genome altogether, but it turns out that actually with some clever referencing, some savvy analysis pipelines, we know actually how to generate pretty good confident genotypes with actually pretty low coverage data in general. And so even with this profile of chromatin, variation assessed by chromatin accessibility, you can actually get an altogether not perfect, but pretty good map of genetic variation across most of the genome in this way, and then use that as your reagent to test for chromatin accessibility QTL mapping. And so that's a very acute, I think it's a very cute idea. The genotyping works pretty well. You can discover CAQTLs that way. All the data is quite heterogeneous, so it doesn't tell you really much about single cell states or the tissue specific states because there's just like, you can imagine all the data that's been generated from ATAC-seq. It's from primary tissues, from cell lines, from iPSCs that are either not differentiated or are differentiated. There's sample overlaps that you have to account for. So definitely pretty complex. But the robust signals, those that have consistent change in chromatin accessibility state across multiple tissue, sample, and individual states, those are probably real QTLs. And this paper does a really good, provides a nice example of a larger comprehensive catalog of those sites that seem to replicate when you compare to other QTL data sets of this type that have been generated. So that's, kind of like opening the door to like collect you use what data that we have to do QTL mapping of this particular molecular phenotype, but still the endeavor proceeds to actually go to primary tissue itself and just build the catalog of CAQTLs within those in those resources. So that's work. That's definitely work ongoing, particularly in the liver. But then you can start to think about cool other things that you could do with the data too. So chromatin accessibility, sort of like I sort of imply this idea that like, oh, you have open chromatin and it allows regulatory architecture, transcription factors to occupy the site and then promote transcription nascently subsequently. What if it turned out that like in the process of generating the attack-seq data, the transposable elements that the transposases that are sort of integrating that you're using to measure chromatin accessibility state actually couldn't get to the site because endogenously there was a transcription factor kind of in the way, so preventing the transposase from actually attacking the site and digesting it. That turns out to actually happen, and it is true. So it's sort of like there's this gap in chromatin accessibility reads that you map to potentially transcription factors that might just be in the way. So they kind of like leave this footprint of a dearth of regulatory reads. And so there actually exist methods that, if you have enough coverage in the data, can actually score the presence of a transcription factor in that kind of like footprinted of a site. So at first, calculating the footprint that a transcription factor that you don't even know about, you don't even know what the transcription factor is. All you're observing in the data is the fact that something must have been there because you see this sort of like coverage difference in the peak of chromatin accessibility that you're mapping. And you can see that if your depth of accessibility is high enough. But once you have that score, you could even further hypothesize, well, what if that transcription factor occupied differently, more strongly or more weakly? More strongly would mean that you would have a further dearth of reads mapping to it, or if it bind more weakly, you'd have more reads mapping to it. And what if that was genotype dependent? So you actually have a genetic variant that's associated with this footprint altogether. And you could imagine a model where that could make sense. If you had if you imagine the sort of canonical motif that transcription factor occupies, and the genetic variant was in fact disrupting or changing that motif away from a stronger motif to a weaker motif, or perhaps creating a stronger motif, so the motif itself was weaker, but the genetic variation change actually makes that motif stronger, then you could easily imagine where that motif, the magnetism that it's drawing either a transcription factor to bind or not bind to that site might be proportional to that genetic variant. So you could actually use genetic variations to map a different type of QTL together, a footprint QTL. So embedded within the region of chromatin accessibility. So I've been involved in this paper, which we try to take that idea to its natural fruition. And it turns out that you can, in fact, map footprint QTLs even in a small amount of QTL data generated primarily from, in this case, from liver. So that kind of opens the door to thinking about other types of larger scale footprint QTL experiments that really help dissect in a way a better understanding of regulatorily what's going on in these particular loci. It would be a disservice if I didn't mention the fact that single cell data is just changing everything about how we're thinking about all the problems that we're trying to map in this space. And it's changing in a way in the sort of like straightforward and obvious ways, which is to say, we've been looking at bulk tissue, and really we know there's cell types in that bulk tissue, and perhaps the disease biology or mechanism is really specific to a subset of those cell types or one specific cell type. So if we could just assay that cell type specifically, we could do discovery better, link our genetic variants to disease in that cellular state, and it would change our mind and influence how we think about our problem. And it absolutely does. And there's a lot of different data types that are being generated we are actively trying to sort this out, collect data with droplet-based capture to get as many cells as we can in as many individuals as we can and in as many known cell type states, but also potentially novel cell type states that we didn't know about because we can profile these things at scale. And it's not just measuring expression. Probably the most exciting, from my point of view, developments in the space is sort of jointly profiling not just one type of molecular phenotype, but multiple molecular phenotypes jointly. So my favorite, I think, is the multi-omics gene expression, transcript abundance with chromatin accessibility state. You can measure in the same cell simultaneously. So that just opens the door to a whole lot of different things you can do, including but not limited to, quantitative trait locus analysis, where you map genetic variants associated with expression or chromatin accessibility at the level of at least pseudo-bulk merged single cell type states. You can map cis-regulatory elements by looking for the cells that have correlation in accessibility and expression state that are coordinated. So that tells you like perhaps CREs that you didn't know about that are specific to that cell type state. And then you can always use all that data to map to our genetic-wide association study hits for your traits of interest, particular to that tissue or cell type, and just really start unmasking much higher resolution state of cell biology to physiological state. So this is obviously transformative. It is transforming all of how we're thinking about these problems. I think it will continue to do so in the next 5 to 10 years, especially as we're collecting a lot more data with a lot more cells across a lot more subjects in a lot more physiologically perturbed states. So whether or not that's across progression of disease, but in different severities of disease across different population groups of all different people. We're just going to learn a whole lot in the next 5 to 10 years on this score. And again, I would also be remiss if I didn't point out the CRISPR revolution in trying to model genetic variation and demonstrate functional relevance of everything we find. So, I am a computational scientist. I will concede that all of the work to sort of like identify genetic variation and map everything, all of that is at the resolution of a computational experiment. It's a prediction and a guess, and hopefully a well-educated guess, but still a statistical guess that you would really like to validate with different types of functional experiments. And, prior to the CRISPR revolution, this was really tedious to do in a lot of scale. But the CRISPR revolution just makes doing these kinds of experiments at scale way more feasible. And we just absolutely have to have that scale. We're identifying, signals of association across a bunch of traits. It's not one signal, it's not 10, it's 100,000, tens of thousands of signals. And many of them are relevant to multiple traits. And we have to pick the right models for all these things. There's a lot of caveats going into it, but it's just, there's a lot of throughput that needs to be generated in these models. And it's important because we definitely want to figure out that like the statistical evidence supporting the right variant, the right tissue, the right mechanism, and the right gene are the right one. If we get any of those wrong, all of the things that we want to do subsequently, either further computational informatics experiments like at a systems level or developing therapeutics or however you want to think about it, all of those things really require us to get as much of those central questions correct and supported by evidence in humans as much as possible before we start making leaps to trying to figure out physiology and systems we can perturb that are a little bit more removed from the central organismal human physiology to a cell state or a cell type or a model system or model cell type or even a model organism that we hope reflects that. But CRISPR makes this easy. There are a whole bunch of experiments at scale, multiple reporter assays, CRISPR-I, CRISPR interference, CRISPR activation experiments, even taking CRISPR at scale to model organisms where you can do this very quickly, like zebrafish or other places. This is all going to transform how we think about and validate ultimately the discoveries we're making from all of these experiments. And again, I think in the next 5 to 10 years, we're already seeing the payouts and fruitions of doing this. This is only going to get better over time. So that's where we are, and that's the thoughts about the future. Thanks for listening, and I hope this was helpful to you in sort of developing your intuition a little bit about human genetics and where we're going. Thanks, and we'll see you next time.